Hello, everyone. Today I'm going to talk to you about bioinformatic approaches to enable low coverage whole genome bisulfite sequencing. And I think the best way to do this is through a case study of some of our recent results investigating Down syndrome neonatal dried blood spots. Down syndrome, or trisomy 21, is the most common chromosomal disorder. It's characterized by distinct facial features, intellectual disability, as well as early onset Alzheimer's disease. There's also immune system abnormalities that result in an increased risk for leukemia, but also a decreased risk for solid tumors, as well as cardiac defects. So by studying Down syndrome, we can learn a lot about not only the above, but also how genetics shapes epigenetics. And this is because Down syndrome is characterized by these trans-acting epigenetic effects. So we see genome-wide alterations to gene expression and the epigenome. So it's not just chromosome 21 that is impacted. And another typical feature of Down syndrome methylation profiles is DNA hypermethylation in a number of tissues. And there's a few candidates for this located on chromosome 21 related to different aspects of establishing DNA methylation, as well as a number of transcription factors. Now, previous 450K array studies have identified these 25 pan-tissue Down syndrome genes based on their differential CpG methylation in multiple tissues, and notably, 24 out of 25 of these are hypermethylated. Previously, we've investigated this and performed the first whole genome bisulfite sequencing analysis of human Down syndrome samples, where we examined human postmortem brains from a select bunch. And we identified over 3,000 DMRs and noticed that about 75% of those were hypermethylated, and that these DMRs had a role in neurodevelopment and metabolism, so very relevant traits to the brain. And what we also saw and replicated was this pan-tissue signal, where we saw many of those hypermethylated single CpGs are actually these very large-scale DMRs overall. In our current study, we're looking at newborn dried blood spots because large banks of these exist in the USA and many European countries. And what's really nice about them is they offer a non-invasive snapshot of very early life before many other events take place that can alter the epigenome. And so there's been a few studies into these, and it's really coming to show that they can provide a lot of insight into many disorders and exposures. However, there hasn't yet been a whole genome bisulfite sequencing analysis due to the low input challenges and the degraded DNA that is inherent to these dried blood spots that have been archived for a long amount of time. However, there have been some studies showing that they can be used for the 450K array. So we took one of these protocols over here that had optimized the DNA extraction and further modified it to go even lower on the input and use that for whole genome bisulfite sequencing. In our experimental design, overall, we had 86 samples of both sexes from 3 groups, and these samples and groups were identified by the California Department of Developmental Services Regional Centers through the CHARGE study. So the 3 groups are Down syndrome. We also have typical development, which was the normal control you would imagine, but also developmental delay. So we have another contrast point that is really unique to our study and will help us get at what distinguishes Down syndrome from other developmental disorders in this heterogeneous group. So these 86 samples were prepared and pooled and sequenced on one NovaSeq S4 flow cell, which is 4 lanes for 150 base pair paired end reads. And we used just 10 nanograms of input genomic DNA after sonication for the bisulfite conversion and library preparation with Swiss Excel NGS MEthylSeq DNA library kit. And this gave us about 5x coverage, which is 100 million reads for all our samples. So we're really happy with the results, and many of them passed QC with flying colors overall. So it's really great to see that this challenging sample source could be assayed by whole genome bisulfite sequencing. Now, in order to analyze low coverage WGBS, our group has been working on a number of novel repositories to enable this that leverage a lot of the state-of-the-art tools and put them together in a nice way. And so the 2 that we've developed are CpGME, which is for the alignment of the data, as well as DMRicher for DMR calling. And these repositories are really optimized for use on a high-performance computing cluster. And they support a diverse range of organisms, including humans, mice, rats, rhesus macaques, chimpanzee, zebrafish, chicken, cow, fly, and Arabidopsis, and they're both available on my GitHub. So please feel free to check them out and email me if you have any questions when you're trying them out. So for alignment, we use CPGME, which is this optimized set of shell scripts that goes from the raw reads, the FASTQ files, and produces the CPG count matrices that are needed. It's largely based on the Bismarck methylation aligner and extractor, and also uses a number of other tools like TrimGalore, FastQ Screen, Picard tools, and MultiQC. So as I mentioned, we'll start with the FastQ files, trim the adapters and the methylation bias that's inherent to post-bisulfite adapter tagging method. It will screen for contaminating genomes to confirm that you are aligning only to your genome of interest. It will also align to your reference genome, remove PCR duplicates, and then calculate the insert size metrics as well as the coverage, extract the CPG methylation, and use it to generate these cytosine reports, which are the genome-wide count matrices of CPG methylation. And then it will also summarize the results by producing quality control and quality analysis metrics, which all go into a nice HTML report at the end, so you can look over all your samples and check out if any have failed. So for DMR calling, we use DMRicher, which is an R package, but also an executable that accompanies it. And it utilizes these CPG count matrices from CPGME to identify DMRs and perform the downstream analyses and data visualization. And it largely utilizes two algorithms, which are DMR-seq and BS-seq. And these are smoothing and weighting-based approaches for the statistical inference of DMRs. And this is what really enables low-coverage WGBS. And these approaches really benefit from more biological replicates over deeper sequencing. So it's much better to have, a handful of samples sequenced at a 5x as opposed to just a few sequenced at 30x. And in the end, you'll get permutation or empirical p-values and q-values for differentially methylated regions and blocks. The DMRs range from a few hundred BP to a few KB and must have at least 5 CPGs with correlated methylation levels. And the blocks are a bit of a different perspective for larger scale change where the smoothing is a bit different and they're typically above 5 KB in size. And what's nice about this approach and the DMR-seq algorithm is that you can also adjust for covariates like sex and age in your analyses. So as I mentioned, what really enables low coverage WGBS are these smoothing and weighting based approaches. So they really allow you to identify larger scale differentially methylated regions. So we're not focused on single CPG sites in these analyses, but rather ones that are closely clustered together and functionally correlated. And in order to identify them without smoothing and weighting, you would need about 25 to 30x coverage. However, with smoothing and weighting, you need only 1 to 5x coverage. And this is because these approaches can infer the methylation level of a lower coverage CPG site from a neighboring higher coverage CPG site. So the key concept here is detecting methylation at 1 out of 2 reads is less informative than detecting methylation at 5 out of 10 reads, even though both of those will tell you that you have 50% methylation. Over here, you can see one of the figures from the early BSC paper. I'm just showing the advantages of smoothing and how it can carefully recapitulate it, where you see the pink line representing 25x coverage, and you can see the methylation levels over here over 1 KB kind of windows. And the pink lines are 25x coverage, and the black lines are this 5x subset. You can see it very closely recapitulates the methylation level by using just a much lower level of reads. So I've put together this approach in all these packages in DMRicher. And so DMRicher will take the CPG count matrices, as well as a design matrix where you specify your samples and the covariates you want to adjust for. It will then filter CPG sites for coverage to make sure you meet certain thresholds. But typically we get over 20 million CPGs that we can assay in all of our approaches where we have between 1 to 5x coverage. It will then call DMRs in blocks. For the DMRs, it will also annotate them, produce an HTML report, perform ontology and pathway analyses, and generate a Manhattan plot. It will also then create these individual smooth methylation values, since the DMR-seq approach really focuses on the difference between groups. And then it will produce some DMR plots, a heat map of your samples, as well as global statistics and principal component analyses. And something we've recently been working on is incorporating a machine learning feature selection. And for the larger blocks of methylation, you also get a list of these blocks and a plot of them too. So The first thing that we looked at when we got our data back from these newborn dried blood spots was the global profiles. And we wanted to really see first that if we could confirm Down syndrome in our samples by WGBS, since this would be a nice quality control step. So we binned the read depth in these 5 KB bins and also normalized the copy number. And what you can see here is very clearly that for the chromosome 21 Q arm, which is within these boundaries, that the Down syndrome samples, there's 3 copies present, but for developmental delay in typical development groups, there's 2 copies present. So the trisomy is only present in our Down syndrome group and not in any of the other groups. So this was a nice quick look to confirm the diagnosis of all our samples that we received. We also noticed something striking, which was when we took on these smooth 20 KB CPG windows for the over 24 million CPG sites that we could assay, that there was a very small but significant shift in methylation where we saw global hypomethylation in our Down syndrome samples over here in this density plot showing percent methylation. Next, we zoomed in a bit more and performed the block analysis. And what we found is that there's 2 significant regions that are actually these known Down syndrome pan-tissue CPG sites. And we've really expanded them here, and it's quite striking to see them. So the first one we see is RUNX1, which is the runt-related transcription factor. Over here, you can see one of the plots where it shows the exons for the genes, as well as the CPG annotations, of whether it's an island, shore, shelf, or open sea. The dots over here are colored by group, where red is Down syndrome, green is developmental delay, and blue is typical development. And each dot represents a CPG assay, and then the size represents the overall coverage. And then we have these smoothed average lines here just to get a nice clean visualization of the coverage and the methylation levels. So what you can see here is that we have a very clear and distinguishing large scale change in the Down syndrome samples, where over 20 KB in sequence is hypermethylated that very clearly distinguishes them from both the developmental delay and typical development samples. And RUNX1 is striking because in addition to being one of these pan-tissue genes, it's involved in the development of blood cells and frequently mutated in acute myeloid leukemia. And it's also located on chromosome 21 itself, which is striking. But what's also interesting about it too is there's a lot of new research emerging that it doesn't just have a role in blood, but also in neurodevelopment. And our second kind of pan-tissue block that we noticed was actually these clustered protocadherins, which I've seen in my research before. They play this key role in neurodevelopment where they actually establish the single cell diversity of neurons. So that's how neighboring neurons of the same type know to connect to each other, but not themselves. And they're this really large cluster of genes, as you can see over here. This is mainly the gamma cluster. And they have this very complex epigenetic regulation with CTCF binding sites in the CPG islands of their promoters that encourages this alternative choice. And as such, they've been implicated in a number of neurodevelopmental disorders and also exposures. So very striking to see that our 2 large blocks have such interesting functional consequences. Next, we zoomed in even more and really started to focus in on the smaller scale DMR profiles. Over here, you can see a heat map of the hierarchical clustering of the Down syndrome versus typical development contrast DMRs. We see that we have over 3,000 significant DMRs. 47% of them are hypermethylated, 53% hypomethylated. So even though we saw this genome-wide hypomethylation, it's still a relatively even split between hypermethylated and hypomethylated DMRs. And these come from about 22,000 background regions that were kind of made up from over 24 million CPGs that we were able to assay in this sample source. So over here, you can see that we can very cleanly distinguish Down syndrome from typical development. Each column represents a sample. The Down syndrome are in red and the typical development in blue. And then each row represents a differentially methylated region, and the clustering is very clean overall in separating the samples at an individual level. We also noticed that we had many of these hypermethylated pan-tissue Down syndrome CPGs, except we were expanding them from just 1 or 2 CPG sites to much larger DMRs that were, a few hundred base pairs to actually several KB in length overall too, which was nice to see. And what we saw is that we were actually able to find mainly the hypermethylated ones, since 24 out of 25 of these sites are known to be hypermethylated. That's what we're detecting, but a very clear signature there with many of the key genes, including the blocks where they've been refined more to smaller scale DMRs, depending on the perspective of zooming, but also the Hox family members, some calcium signaling, and quite a few others related to development. Finally, even though we had over 3,000 DMRs, we wanted to perform machine learning feature selection to see if we could refine this list further, since this might be ideal for future follow-up studies and creating a smaller panel to sequence the key areas that we're finding. So in this approach, we take the top common DMRs identified by both the random forest and support vector machine learning algorithms. And over here, you can see a heat map of the clustering of the samples. Now, instead of z-scores of the methylation here, we're actually just plotting the percent methylation. But what you can see very clearly is that the Down syndrome and typical development cluster very separately, just based on about a dozen DMRs. And notably, we find that the RUNX1 block is actually one that has been selected by the machine learning algorithms. So we also did the contrast of Down syndrome to developmental delay. And what we found is that there's over 2,000, almost 3,000 really significant DMRs, very similar here with a relatively even split between hypermethylated and hypomethylated, coming from over 20,000 background regions and over 24 million CPGs that we could assay. And over here, you can see the clustering where the green is developmental delay and the red is Down syndrome. So very similar to the contrast of typical development overall. And the next, which kind of serves as our sanity check to show that everything's going right, is this comparison of the developmental delay to typical development, just because the developmental delay is very heterogeneous overall. So we don't expect to nearly see as much of a strong signature as we saw in Down syndrome. That's precisely what we're seeing here. We only get 59 significant DMRs, but none of them pass the extra FDR correction overall. And we see a relatively even split of hypermethylated and hypomethylated too, although a bit more of a shift to hypermethylated. And what we're noticing is there's much fewer background regions because there's really not a lot of change to test. And this is still coming from over 25 million CPGs that we could assay. What you'll notice is this heat map does look very different overall from the other Down syndrome contrast, just because it's not able to cluster the samples as well and really reflects the heterogeneity of the developmental delay. So what we noticed when we kind of confirmed from the literature is that these DMRs do show a genome-wide distribution. So even though chromosome 21 is the extra chromosome, we see these transacting effects across the genome. You can see them for the Down syndrome versus typical development, as well as Down syndrome versus developmental delay. And then, of course, the contrast here of DD versus TD. And over here, the Manhattan plots generated by our pipelines, where it shows the chromosome at the bottom over there, where you have all the chromosomes, as well as a track of the density, as well as the negative log 10 p-value. So the more significant the site, the higher up it is overall. Next, we wanted to really look and see how the DMRs from all these different contrasts overlap and get at a core signature. So over here, you can see an overlap based on genomic coordinates. So if any of the DMRs directly overlap, not just by their gene symbol, but actually by their physical coordinates, then they'll be counted as an overlap. And what this Venn diagram of the contrast show is that for the Down syndrome contrast, the majority of the DMRs between them are actually shared regions overall, which is nice to see. And just to get another perspective on that from genomic overlaps, we're going by a KB sequence now rather than just any overlap. And what we can see is that when we compare all three contrasts, there's very little overlap on this upset plot. However, the Down syndrome contrast show the largest amount of unique sequence overall between each other. And of course, there still is some unique sequence there, but very nice to see that most of the DMRs we're identifying really are shared across the Down syndrome contrast. And so then we took this list of consensus DMRs, merged them together if they overlap to refine them a bit more, since we didn't want to double count anything. And this gives just over 4,000 DMRs. And as you can see by both the heat maps and the principal components analysis, is that these DMRs can very cleanly distinguish the Down syndrome samples from the developmental delay and typical development samples. So after that, we want to perform some enrichment testing and see exactly what these DMRs were up to. So first, we looked at the genic annotations over here. What we noticed is that for most of these annotations, like enhancers, promoters, introns, exons, we actually see a de-enrichment in the Down syndrome contrast. However, we do notice that we have a significant enrichment for 5' UTRs in the Down syndrome contrast, and really no signal coming out of our other developmental delay versus typical development contrast. So this is very interesting to see, since it's novel in the literature, and it could actually indicate a role of alternative promoters, and it's something we're looking into further right now. Next, we also wanted to look at the CpG annotations, which are very typical of the Illumina arrays that we always like to compare to. And what we found is that for our contrast, we see significant enrichments for CpG islands overall, and de-enrichments for the other features. So it seems like a lot of these DMRs are very focused on the CpG island itself rather than the outside sequence flanking it. Next, we performed gene ontology analyses to really get an idea about the functional consequences of these genes in addition to where in the genome they're landing. And what you can see over here is a bar chart of the negative log 10 p-value of the enrichment. So what I've done is I've taken the enrichments from the Down syndrome versus typical development contrast and took the top terms from there ranked by the p-value and then pulled out the corresponding terms for the other two contrasts to see how they compare. And so what we see actually is that there's an enrichment for ontologies that we saw in the brain, like neurodevelopment and metabolism. So this might reflect some of that pan tissue signature. But what was also unique to us really is that we see a new ontology that hasn't really been implicated yet, which is transcriptional regulation. A lot of genes really seem to be regulating gene expression overall, and that's really unique. And it's also interesting still to see that we see a lot about neurodevelopment in these newborn blood spots. Next, I performed my own Down syndrome pan tissue analysis and leveraged a bunch of the 450K studies. So what I did is I extracted the CpG sites that were significant from a lot of these studies, typically 450K, but also some of the other reduced representation methods, as well as our previous study of the adult frontal cortex. So I compared to the enrichments based in other studies, so what they found to be different between Down syndrome control and their respective tissue, and saw how well my DMRs would overlap. And I also split them by hypermethylated, which is in red, as well as hypomethylated in blue. And you can see each facet represents a different contrast for Down syndrome versus typical development versus developmental delay, and as well as our sanity check of DD versus TD. So what we see is that the strongest enrichments really are in the tissue that we're assaying, which is blood. You know, the sorted adult peripheral T lymphocytes are the top hits, but we also have the whole blood DMR CpGs, as well as another study that looked at neonatal blood from a handful of samples. And what's also interesting to note is that the enrichment is very kind of cross-tissue overall for the hypermethylated DMRs in red. A lot of different tissues are showing up as very significant. However, when we look at the hypomethylated DMR, significant enrichments are really only seen in the blood tissues. And actually, the further down we go, we start seeing significant de-enrichments, particularly for the brain. So even though the hypermethylated DMRs significantly overlap, the hypomethylated ones are actually much less present than when you would expect by random chance. And this starts to get at some of this difference between the hypermethylation being pan tissue and the hypomethylation actually being much more tissue specific. And that's something that we've grown very interested in. So I also compared our data to the roadmap Epigenomics 127 reference epigenomes and looked at their core histone modifications. So over here are the enrichments for the histone modifications at the hypomethylated regions for the Down syndrome versus TD analysis, but Down syndrome versus DD was also very similar, and there's really no pattern in our sanity check analysis. So what you can see here overall is the index here represents the different tissues and the enrichments for them, so a bit different than your typical heat map. And what we notice is that H3K6ME3 actually shows a very kind of, cross-tissue enrichment. So this mark doesn't really seem to be as tissue-specific, but when we look at H3K4ME1, we actually see very strong tissue specificity for it in the enrichment overall, and it's very specific to a subset of the blood samples there. So this really indicates tissue-specific H3K4ME1, as well as H3K4ME3 shows a similar pattern, although not as striking. And what's noticeable about this is that this is actually very similar to the pattern we had seen in our brain samples as well. So these hypomethylated DMRs really are starting to reflect something tissue specific. We want to look a bit further into this, so we performed some transcription factor motif analysis and broke it up by hypermethylated and hypomethylated. So what you'll see is that many of these different motif families do show significant enrichments, but the most striking in the top one in all of them is related to the runt family, and very specifically in the hypomethylated DMRs for our Down syndrome contrast, but not in our relatively null contrast overall. And so these heat maps are ranked by the negative log p-value, and the stars indicate if they pass the FDR or not. So what's noticeable though is the runt family, the key member here and the one driving it is actually RUNX1. And this is the top motif, and both our Down syndrome contrast and really starts to give a bit more support for this tissue specificity of the hypomethylated DMRs. And just to refresh your memory, this was one of our two significant pan-tissue blocks. So it really seems like this region showing a large change is really starting to be behind a lot of that hypomethylation we're seeing. So we're very interested in further understanding this, and to really summarize that hypomethylation profile, we see this slight reduction in global methylation where there's DS pan-tissue specific enrichments for only blood for the hypomethylated regions, as well as this blood-specific H3K4ME1 enrichment and this enrichment for the RUNX1 transcription factor motifs. So mechanistically, there has been some studies into RUNX1, and what they're finding is actually that RUNX1 actively demethylates the DNA it binds to during blood development by recruiting DNA demethylation enzymes like TET2, TET3, TDG, as well as GAD45. So a very kind of clear mechanism is starting to emerge from our relatively more associative analyses. And because RUNX1 is on chromosome 21 and is overexpressed, we're hypothesizing that this block of hypomethylation we're seeing is either a response to attenuate the overexpression that's demethylating a lot of the genome, or it could also be driven by overexpression of DNA methyltransferase 3L, which is also on chromosome 21 and is something we're actively exploring now. So in terms of our conclusions and take-home messages, we've confirmed and expanded on the DS pan-tissue gene hypermethylation. So it's nice to see this confirmation when applying a new technology and able to recapitulate a lot of what's been found on the arrays. And we've also discovered this potential novel role for RUNX1 in the DS tissue-specific hypomethylation. And in terms of big picture, we're really, the take-home message here is that we can now actually assay the entire methylome of newborn blood spots by whole genome bisulfite sequencing, which is great since we have a number of projects for many different disorders planned for this. And really starts to show that low coverage WGBS of low input samples can provide big insight. So I'd like to thank everyone in the LaSalle Lab, as well as our collaborators and the funding agencies for supporting this research. And thank you as well for listening.